Hello everyone, very warm welcome to Sri Chaitanya Academy. My name is Kiran Chaudhary and today we're going to do discussion on your last grand test paper. So let's get started. The first question. Yes, everyone present in this class, give me a thumbs up if you're listening. Let's start. And also let me know which particular option you have given as the answer. So let's make this quite engaging and fruitful for you as well. Which one of the following algae forms pear-shaped gametes? Is it volvox? No. Spirogyra? No. Chlamydomonas? No. Fucus? Yes, because it is the brown algae which form pear-shaped gametes. Very good. Answer is 4, Fucus. Clear, guys? Very good. Yes, option is 4. Good evening everyone. Let me know what is the answer to the next question in the chat box. Algin and carrageen are the hydrocolloids. Algin and carrageen, my dear students, are hydrocolloids that are obtained from dash and dash algae respectively. So please notice algin is obtained from your brown algae and carrageen from red algae. So answer should be 3. Algin from brown, carrageen from red. Correct answer is 3. Yes everyone? Very good. Yogamantham Reddy got 720. Very nice. All the best everyone present in this class for your NEET exam. All the very best. Next. So here the answer is 3. Which of the following cells help in maintaining pressure gradient in sieve tubes? Very obvious answer, and that is your companion cell. So correct answer is 2, my dear students, companion cells. Companion cells helps in maintaining pressure gradient in sieve tubes. Answer is 2. Next, let me know which particular option you have given answer over here. Yes, everyone, participate. Let me know your answers here. Which of all the following have cells? All the following cells have nucleus at maturity except. So which one out of the 4 does not have cell? Does not have nucleus in the cell at maturity. So if you see over here, my dear students, the very first option, sieve tubes. So sieve tubes will not possess nucleus at maturity. So your correct answer is 1. Next, very good. List. Match list 1 with list 2. SER. So smooth endoplasmic reticulum, responsible for or involved in glycogen metabolism. Lysosomes, suicidal bags. Nucleoid, you will see in bacteria. Chromatin will be present in the nucleus. So 2, 3, 4, 1. 2, 3, 4, 1. So 2, 3, 4 and 1. So 1 should be your right answer. Right guys, 1 should be your right answer. Excellent. Yes, now let's see the next question. Which layer of the cell envelope in prokaryotes prevent them from bursting or collapsing? Very simple. Answer is cell wall. Answer is 4, cell wall. Next. Meiosis 1 is the actual reductional division where the reduction in chromosome number at each pole due to disjunction, and this disjunction is observed in anaphase 1. Very good. Anaphase 1. Correct answer is 1, anaphase 1. Yes, excellent. Next question, guys. Bacterial and fungal enzymes degrade detritus into simpler inorganic substances. This process is called as catabolism. Answer is 3, my dear students, catabolism. Clear everyone? Excellent. Next. Electron donor to PS2 in non-cyclic electron transport is? Water. Very, very simple. Answer is 1, guys. Water. Clear? Next. Read the following statements with respect to glycolysis and select the correct option. तो बताइए इसका correct option कौन सा होना चाहिए, guys, everyone? With respect to glycolysis. So, it takes place in cytoplasm. Yes, glycolysis takes place in cytoplasm. It is complete oxidation of glucose. No, it is not a complete. It is a part of aerobic respiration only. So, because of this only word, no. It occurs in all living organisms. This is correct. So, A is correct and D is correct. So, your correct answer is A and D only. So, fourth is your right answer. A and D only. Right, guys, everyone? Very good. Next question. Biological control methods adopted in agricultural pest control are based on the ability of the predator to regulate the prey population. Absolutely correct from NCERT. Competition between species can never occur in a habitat where resources are unlimited. This is wrong. Even when the resources are abundant, the competition can occur. So, this is wrong. So, statement 1 is correct, but statement 2 is incorrect. So, your correct answer is 2, guys. So, your correct answer is 2. Next. Which of the following is the main cause of the extinction of stellar sea cow? The answer is 1, over-exploitation. Answer is 1, over-exploitation. Very good. Excellent. Next question. Interesting question, but very simple. 4 microspore mother cells after meiotic division, that means meiosis, in angiosperms will form. So, you can just remember for 1 microspore mother cell and you can multiply by 4 for the result of 4 microspore mother cell. So, each and every microspore mother cell which will undergo meiosis to produce 1 microspore tetrad which will have 4 microspores which will develop into 4 pollen grains. So, simple. So, total 4 microspore mother cell after meiosis will give 4 microspore tetrad or 16 pollen grains. Simple, very, very simple. Answer is 4. Next. Which of the following is caused by a chromosomal disorder involving autosome? So, here is your answer. Involving autosome. Very simple. Down syndrome. So, your correct answer is 2. Down syndrome. Yes, excellent. Next question on your screen. In the typical Mendelian's monohybrid cross, law of dominance is used to explain the expression of both the parental characters in F1. No, that was even not the case. The purity of gametes? No. Expression of only one of the parental characters in F2? This is also incorrect statement. The proportion of 3:1 phenotypes, that means 3 out of the 4 were tall and 1 out of the 4 was dwarf. Example, just an example I am taking, okay. So, the proportion of 3:1 phenotypes obtained in F2 generation, absolutely correct. Explained by Mendel's law of dominance. Next, answer is 4. How many true breeding pea plant varieties were selected by Mendel? In such question, please focus. Is it asking about number of true breeding pea plant varieties, or it is asking about the number of contrasting characters that he chose? Answers will be different, okay. So, this time they are asking how many true breeding pea plant varieties. So, true breeding pea plant varieties were 14 in number. So, answer is 4, guys. Answer is 4, that is 14. Clear? Excellent. Next question. Okay, that was an interesting question, but very simple. Many questions like this we have seen in the previous NEET exam. So, to solve this question, if you see what is given, which of the following DNA sequences lead to the formation of the below given mRNA. So this is the sequence of mRNA, bacha. This is the sequence of produced mRNA through the transcription process, okay, through the transcription process. So in the transcription unit, if you notice, a segment of DNA will participate, and from that segment also, my dear students, only one of the strand, which is called as template strand, will get copied, and the polarity of the template strand, my dear students, is going to be 3:5. And you have to know three things over here. First, the polarity of the template strand is going to be 3:5. Second thing, you have to use the complementary base pairing rule. And the third thing, notice in mRNA you will see uracil, but in DNA instead of uracil you will have thymine. So if you know these three things, easily you can get the answer. One is the right answer here. Okay, one is the right answer here. Yes. Very good. Which of the following occurs in the cytoplasm of the eukaryotic cell during S phase? There were two important key points here, or keywords here. First is during S phase and second is in the cytoplasm. So only the activities which is happening in the cytoplasm during S phase you were supposed to answer. So replication of a DNA, yes, during S phase, but not in cytoplasm of the eukaryotic cell, rather in the nucleus of the eukaryotic cell. So this is also wrong. Then replication of RNA, no. But the B statement says duplication of the centriole, yes, in the cytoplasm during S phase. So B only is your right answer. So answer is 2, B only. Clear, guys, everyone? Next. AR. DNA polymerase can synthesize DNA. DNA polymerase can synthesize DNA in 5:3' direction. Correct. DNA polymerase require a free hydroxyl OH group on the 3' of the growing strand to catalyze phosphodiester bond formation with the 5' phosphate of the incoming nucleotide. That's absolutely correct. And also explaining the assertion accurately. So here both assertion and reason are true, and reason is the correct explanation of the assertion. So the correct answer is 1 over here. The correct answer is 1. Clear? Good. Next question. Come on, guys, participate. Let me know your answers. Next question. Very simple. You have to find incorrect match. So if you see Swiss cheese, Propionibacterium shermani, correct. Penicillium, through Penicillium notatum, correct. Cyclosporinate, Trichoderma polysporum, correct. But statin through Saccharomyces cerevisiae, that is wrong. Statin is through Monascus. So this becomes the answer. This is the incorrect match. So select the incorrect match. The fourth statement is your, or fourth is the incorrect match. Answer is 4. Okay, answer is 4. Next, very good. In which of the following options the two taxa given are correctly matched? So if you see over here, my dear students, Triticum and Convolvulaceae for the wheat, no. Diptera and Canidae for the housefly, no. Musca and Anacardiaceae for mango, no. Next is Chordata and Hominidae for man. This is absolutely correct. So your correct answer is 4, guys. So your correct answer is 4. Yes, very good. Next. Excellent, excellent. So your correct answer for this question is 4. Which of the following protists show both autotrophic and heterotrophic? So both autotrophic and heterotrophic means mixotrophic nutrition. So it will be seen in Euglena, which belongs to euglenoids. So answer is 4. Answer is 4, euglenoids. Clear, guys? Very simple. Next. In hypogynous flower, the other parts of the flower will rise below the level of the gynoecium. Yes, that's right. That's how we define a hypogynous flower. The ovary which is present in peach is said to be inferior. No, it is half inferior. Plum, peach and rose, half inferior. So statement 1 is correct, but statement 2 is incorrect. So statement 1 is correct, but statement 2 is incorrect. So 1 should be your right answer. 1 should be your right answer. Clear, guys? Very good. Excellent. 1 is your right answer. Statement 1 is correct, but statement 2 is incorrect. Next question on your screen. Yes, everyone try to answer. In dicot stem, vascular bundles are collateral, conjoined and closed. In monocot stem, vascular bundles are collateral and open. Monocots, open. So statement 1 and statement 2 both are incorrect. In the light of the above statement, choose the correct answer. Both statement 1 and statement 2 are incorrect. So answer is 4, guys. Both statement 1 and statement 2 are incorrect. So your correct answer is 4. Yes, everyone? Excellent, excellent. Yes, that is also right, Vrishti. Absolutely right, absolutely right. This is also. Yeah, excellent. Answer is 4. Split gene arrangement is characteristic of eukaryotes. Yes, split genes are found in eukaryotic DNA. Prokaryotic genes possess both introns and exons. No, this is wrong. So split gene arrangement is a characteristic of eukaryotic genes. Absolutely correct. Prokaryotic genes also possess introns and exons. No. So statement 1 is correct, but statement 2 is incorrect. Answer is 1. Answer is 1. Clear, guys? Answer is 1. Yes, excellent. Answer is 1. Next. Number of eggs or egg in each ovule of the flowering plant. So it has to be 1. Egg या फिर आप female gamete भी consider कर सकते हैं. Yes. Next. So question number 38. Number of egg or eggs in each ovule of the flowering plant is or are? Answer is 1. Next question. Now listen this carefully. List 1 and list 2 you have to match. Codominance. Codominance, AB blood group is the example. Incomplete dominance. Flower color in dog flower in heterozygous condition. Incomplete dominance. Polygenic inheritance, human skin color. Linkage. Genes controlling eye color and body color in Drosophila 3. So 4, 2, 1 and 3. 4, 2, 1, 3. 4, 2, 1 and 3. So 3 should be your right answer. Yes, guys, 3 should be your right answer. Clear, everyone? Next. Pteridophytes are limited and restricted to a narrow geographical region. Yes, line of NCERT. Prothallus in pteridophytes is multicellular, free-living, mostly photosynthetic, thalloid gametophyte, which require cool, damp and shady place to grow. This is also line of NCERT, absolutely correct. And yes, because of which pteridophytes are limited and restricted to a narrow geographical region because of the specific requirements. So yes, both assertion and reason are true, and reason is the correct explanation of the assertion. Answer is 1. Clear, guys? Answer is 1. Excellent. Next question on your screen. So here answer is 1. Okay, choose the correct statement about the plant. So if you see, this is also from your NCERT, this diagram. This is of Funaria and it is showing the monoecious gametophyte with attached sporophyte. This is your gametophyte with the attached sporophyte. So answer is 3. Okay guys, this is Funaria having a monoecious gametophyte with attached sporophyte. S for sporophyte, G for gametophyte. Answer is 3. Yes everyone? Excellent, excellent. Answer is 3. Repressor protein in lac operon binds to. You can't even expect an easier question than this from lac operon. So, repressor protein in lac operon is going to bind to the operator. It is going to bind to the operator. Answer is 1. Yes, very good. Answer is 1. Okay, multiple statements are here. Let's read them out. Biodiversity is uniform throughout the world. No, it is not. Even distribution is not there. Species inhabiting tropical latitudes had a long evolutionary time. This is right. Next, Alexander von Humboldt observed that within a region, species richness increased with increasing explored area without any limit. No, after a certain region, won't be that much effective. Wrong. David Tillman showed that in his experiments that increased diversity contributed to a higher productivity. This is in fact correct. So what is correct? B is correct, D is correct. So B and D answer should be 4. Okay guys, so correct answer is 4. B is correct and D is correct. So correct answer. Here you were supposed to find out, mark the options that include only correct statements. So which of the following are correct? B is correct, D is correct. B and D answer is 4. Good, excellent. Next. If the female parent bears, your considered female parent plant, bears a bisexual flowers, then emasculation is required during artificial hybridization. Correct. Process of bagging is necessary for the emasculated flower to prevent contamination of its stigma from unwanted pollen, your undesirable pollen. Yes, both the statements are correct. So both the statement 1 and statement 2 are correct. Answer is 3 guys. Right? Answer is 3. Both the statement 1 and statement 2 are correct. Answer is 3. Clear? Yes. Excellent. Next. Choose the correct option that indicates the sequence of different stages that occur in the logistic growth of a population. Okay? Yes, sigmoid one. So it will start with lag, followed by acceleration, deceleration and asymptote finally. Right? So answer is 2. Lag, acceleration, deceleration and asymptote. Answer is 2. Your correct answer for this question is 2. Yes everyone? Pretty simple? Excellent. Next question. Which of the following is an autosomal, autosomal dominant genetic disorder given in your NCERT? Very simple. First option, myotonic dystrophy. So the correct answer for this question is 1. Myotonic dystrophy is the autosomal dominant genetic disorder. Next question on your screen, guys. Read the following statement and choose the correct one. Correct one regarding transcription. In eukaryotes, only one type of DNA-dependent RNA polymerase transcribes all type of RNAs? No. This is correct for prokaryotes. In transcription, only a segment of a DNA and only one of the strand of the DNA is copied into RNA. This is correct. The DNA strand that has polarity 5:3 act as a template. No, the DNA strand that has polarity 3:5 act as a template. This is wrong. In transcription unit, promoter is located at the 5' end of the template strand. Remember, in the transcription unit, these positions are given with respect to the coding strand. So, in transcription unit, the promoter is located at 5' end of the coding strand or structural gene if you say is correct. But just saying template strand is wrong. So, this is also wrong. So, your correct answer is 2. Your correct answer is 2. Yes, guys, everyone? Excellent, excellent. Let's see the next question over here. So, the correct answer is 2. Different pathways are followed by the plants in response to environment or phases of life to form different kinds of structure. This ability is called as plasticity. Your answer is 1. Find the incorrect match. Find the incorrect match with respect to placentation. Exile China rose correct. Basil in sunflower correct. Free central and argemone incorrect. So this becomes the answer to your question. Parietal mustard is correct. In fact, in argemone also it will be parietal. So answer is 3. Your correct answer is 3, guys. Yes? Very good. Excellent. So for this question, your correct answer is 3 because they are asking about incorrect match with respect to placentation. Next. Okay, in C3 plants, Kelvin cycle takes place in C3 plants they are asking. Kelvin cycle takes place in mesophyll cells, but in C4 plant, it takes place only in the bundle sheath cells. Correct. Photosynthesis in C4 plant, photosynthesis in C4 plants is not inhibited by oxygen. Yes. In C4 plants have high temperature optima than C3. This is also correct. C4 plants can withstand drought or water stress condition. This is also correct. So all A, B, C, D are correct. So answer is choose the correct answer. So A, B, C and D are correct. So your correct answer is 3. A, B, C and D are correct. Clear, guys, everyone? Yes. Next question. Match. Synapsis, if you see, my dear students, it will happen in prophase 1. Splitting of centromere, anaphase 2. Nuclear membrane and nucleolus reappear, telophase 2 you can go for here. Microtubules from the opposite poles attached to the kinetocores of the homologous chromosome. It has to be the metaphase. So here metaphase 1 is mentioned. So if you see over here, my dear students, answer should be 4, 2, 5 and 1. 4, 2, 5 and 1. So 1 is your right answer. 1 is your right answer. Clear? 1 is your right answer. Next. Incorrect statement. The mass of the living material present at a particular time in a trophic level is called as standing crop. This is right. Vertical distribution of different species occupying different levels is called as stratification. Correct. Identification and enumeration of plants and animal species of an ecosystem gives a species composition. Correct. Organisms at each trophic level always depend upon those at the higher trophic level for their energy demands. No, they depends on the lower, right? So a higher trophic level organism eats the one which is at the lower trophic level. So organisms at each trophic level always depends upon those at higher trophic level for their energy demand. This is wrong because of that higher word. So this is wrong and therefore this becomes the answer. So your correct answer is 4 because they are asking about choose the incorrect statement. So answer is 4. Correct answer is 4. Clear, guys? Next. Very good. Answer is 4. Inner layer of cell wall of pollen grain is made up of. So they are asking about the intine. Remember, pollen grain has 2 wall layers, exine and intine. Exine is the outermost, intine is the inner one. So the inner layer of the cell wall, that means intine of the pollen grain is made up of cellulose and pectin. Intine is made up of cellulose and pectin. So your correct answer is 2, guys. Correct answer is 2, cellulose and pectin. Right? Next. Given below are the two statements, okay, assertion and reason. In Drosophila, the inheritance of eye color is sex-linked. That's right. In Drosophila, genes that control eye color are located on Y chromosome. No, they are on X chromosome. So this is wrong. So statement 1 is true, but statement A is true, but the reason wala statement is false. So the correct answer is 3. Correct answer is 3, guys, because assertion is true but reason is false. A true, R false. Answer is 3. Next question. Excellent. Short sequences of DNA in the human genome that can be used to identify genes that are expressed as RNA, termed as ESTs, express sequence tags. Answer is 1. ESTs. Answer is 1. Next question. FMN, FES are associated with which complex in mitochondrial ETS? Very simple, complex number 1. So your answer is 2. Complex number 1. Answer is 2. Next question. Identify the correct statement function of the different types of RNAs. It is a genetic material in bacteriophage lambda and 5174. No, it's the DNA. It is recessive. RNA receives genetic information from DNA for synthesis of protein. This is right. It is a structural part of ribosomes. Right. It carries amino acid to ribosomes. This is also right. It plays a catalytic role during translation and splicing. Again, correct. So B, C and D are correct, but A is incorrect. We are asking about correct function of different types of RNA. So if you see over here, only B, C, D and E. So your correct answer is 3. Your correct answer is 3. Only B, C, D and E are correct. Correct answer is 3, guys. Clear? Next. Exploring molecular, genetic and species level diversity for producers of economic importance is called as? It is called as bioprospecting. Answer is 1. Bioprospecting. ZW type of sex determination is found in birds in which females are heterogametic. Answer is 4. Birds in which females are heterogametic. Answer is 4, guys. Next question on your screen. Which of the following plants produces highly poisonous cardiac glycosides? Very obvious, Calotropis. So your correct answer is 2, my lovely kids, that is Calotropis. Next question. Okay, read the following features. Cell wall is generally made up of amino acids and sugars. Membrane bound cell organelles are absent. That means it's a prokaryote. Nucleoid or incipient nucleus is composed of naked DNA, RNA, non-histone protein, again indicating the prokaryote. Majority are heterotrophs, that is they depend on other organism or on dead organic matter. They all are talking about prokaryotes. Agar hum kingdoms ki baat kare, which was the prokaryotic kingdom? Monera. So with respect to 5 kingdom classification, the given feature are most likely associated with the members of the kingdom Monera. The correct answer is 1, my dear students, that is Monera. Next question. An example of leaf modification is very simple. Phyllodes of Australian acacia. Phyllodes of Australian acacia. So your correct answer for this question is 2, my dear students, that is phyllodes of Australian acacia. Now we will be taking the last question here. Match list 1 with list 2. Morris-Wilkins X-ray diffraction data of DNA. Taylor, semi-conservative nature of chromosome replication. Marshall-Nirenberg, cell-free system for protein synthesis. 4. Griffith, transforming principle causes transformation. 1. So 3, 2, 4, 1. 3, 2, 4 and 1. So 1 should be your right answer. 1 should be your right answer. So that's all, my dear students. From my side, now the questions from zoology will be discussed. Bye-bye. Take care. All the very best for your upcoming NEET exam.
Hello, very good evening everyone. Now it is time to discuss zoology portion with me. So I hope you all are ready. Hello Vrishti, Manasa, Rehan, Arpita, everyone. So let's see what is the first question that we are going to discuss. So चलिए देखते हैं first question को. Right, study the following statement. Calcium ion play a very important role in blood clotting. Definitely, the plasma of a person of the AB blood group contain both anti-A and anti-B antibody. So when we talk about AB blood group, they have both the antigen on the RBC but no antibodies in the plasma. WBC are the most numerous of all the formed elements in the blood. No, plasma without clotting factor is called serum, which is right. So A and D यहाँ पर right है, correct ही हमको बताना था. So the correct answer will be 3. Yes, C. Very good. Keep it up. Next. Enzyme that catalyze the removal of group from substrate by mechanism other than hydrolysis will be lyases. ठीक है, other than hydrolysis पूछा है, so answer is 2. Now statement 1. Gift is suggested for a woman who cannot provide suitable environment for fertilization. So definitely gift cannot be suggested to them. Gift is the method of transfer of zygote or embryo up to 8 blastomere stage to the fallopian tube. Zift means zygote intrafallopian transfer, so that is true. So 1 is incorrect, but 2 is correct. Correct answer will be, that's why 2. Very good, keep it up, very nice. Keep on telling me the answers. Which of the following is not a cranial bone? Frontal, lacrimal, parietal, occipital. Lacrimal is a facial bone, okay? So it is 2. Next. According to the Big Bang theory, after the initial expansion and subsequent cooling of the universe, galaxies of the present day universe were formed by condensation of gases, expansion of gases, diffusion and precipitation. Definitely it is condensation of gases. Okay, so answer is 1. Remember 1. The volume of air that remain in the human lung at the end of normal exhalation is about. That is functional residual capacity. Okay, so functional residual capacity, okay, that will be the air that remains after normal exhalation. That will include the expiratory reserve volume and the residual volume. So the answer will be 1, that is 2300. Okay, simple question, don't make a mistake here. Who noted that the embryos never pass through the adult stages of the other animal? Direct question taken from NCERT. Answer will be 1. Bears. That is 3. Okay, match. Let's match it. Ribs. Okay, ribs articulates with the thoracic vertebra. Humerus articulate with the glenoid cavity. Femur articulate with the acetabulum, and occipital condyle articulate with the atlas, right? So correct answer will be 3. Next one. Assertion says vaccination help in a quick and massive production of antibodies on subsequent exposure to the particular pathogen. Yes. Vaccination generate memory B cells that recognize the pathogen quickly. So assertion is true, reason is true. Reason is the correct explanation of assertion. So that's why the answer will be 1. Easy question. Diacetylmorphine is commonly called as SMAG. Okay, so diacetylmorphine is heroin, which is also termed as SMAG. 3. Okay, not coke. Okay. Now, RNA interference takes place in all the organisms, prokaryote and eukaryote, as a method of cellular defense mechanism, which is wrong. Restriction endonucleases are used to join vector DNA to desirable gene in order to produce rDNA. No, we use DNA ligase. The vector DNA and the source DNA are cut with the same endonuclease to obtain sticky ends, that is required. Disarmed pathogen vectors are also used in transfer of alien gene into the host. Yes, so C and D will be the correct answer. So answer should be 3. Okay, answer should be only A and B, because we are asked about the incorrect statement. So answer will be 1, because only A and B are incorrect, rest 2 are correct. Okay, next. Which of the following belong to mollusca? So here I can see it is devilfish. Okay, devilfish is octopus, which is mollusca. Okay, so it is 2. Next. Statement 1 says concentration gradient exists across the axonal membrane. That is important. The extracellular fluid contains relatively more sodium ion and fewer potassium ion compared to the exoplasm. Remember, that both the statements here are correct. Okay, so answer should be 1. Very good. Which of the following sexually transmitted infection is completely curable if detected early? So we will look for bacterial or we will look for protozoal infection. So HIV, genital herpes, hepatitis B will not be cured. So it is trichomoniasis. Right? Good. Stirrer in simple stir tank bioreactor has been designed for availability of oxygen throughout the bioreactor and even mixing of the content. Right? So it should be 4. Yes? Very good. Which of the following receive bile from the gallbladder? That should be duodenum. That means 3. Right? Which of the following is not common in snake and lizard? Definitely 2 pairs of limbs not present in snakes. So it is 4. Easy question. Study the following statement and figure out the correct option. Areolar tissue is present beneath the skin. Yes, areolar adipose beneath the skin. Collagen, elastin fiber are absent in blood. Yes. In both bone and cartilage, the cells are located in small spaces called lacuna. Yes. Dense, irregular connective tissue is present in the skin. Yes. The muscle fiber in smooth muscle are held together by cell junctions. Yes. All are correct. So answer should be 1. Yes. Good. So again, we have as the following question. Lipus loop, okay, promotes the phagocytosis of sperm. Multilode 375 is copper releasing IUD that will be matched with. Suppresses the sperm motility, injectables and implants. Okay, inhibit ovulation and implantation, and Saheli inhibits implantation. Okay, so correct answer here should be 2. Right? Got it? Next. Which of the following is not related to the atrial systole? So when atrias are contracting, remember that there will be 30% of blood flow from atria to the ventricle, which is also termed as active filling. So bicuspid and tricuspid valve should be open. So which is not related to the atrial systole is the closure of bicuspid and tricuspid valves, because it should be open. Yes or no? So it is 3. Next. Select the incorrect match. Starch can hold iodine molecule. Yeah, that's why we are getting this blue color. Cellulose contain complex helices. Not at all. Non-essential amino acid can be produced by our body. That's why they are non-essential. We don't need it in our diet. Adenine in substituted purine? Absolutely. So correct answer here should be 2. Very good. Now read the statement and let's see which is the correct statements. The primary oocyte and the first polar body are diploid. Primary oocyte is diploid but not the polar body. The cells released during spermiation and ovulation are haploid. So yes. Morphological differentiation of gamete occur after the completion of meiosis in both spermatogenesis and oogenesis. So differentiation is not happening in oogenesis. So I think this should be wrong. Initiation of both spermatogenesis and oogenesis require a significant increase in the amount of GnRH. Yes, that is true. So the polar body consists of autosomes and the Y chromosome. So only Y chromosome will it be there? No. So we have B and D as the correct statement. So answer should be 1 or A. Perfect. So again we have 3 statements and we need here the correct set of statements. Primary metabolite have identifiable function and play important role in human physiology? Yes. Morphine and codeine are secondary metabolite that are lectins? No, that is concavalin. Okay, lectins are concavalin A. These are alkaloids, yes or no? Many of the secondary metabolite do not have identifiable function and are useful to human welfare, and some have ecological importance also. So A and C will be the correct statement. So answer should be 1 again. Perfect, very good. Next. Alpha-1 antitrypsin is used for the treatment of emphysema. So answer is 4. Next. Which of the following hormone attains its peak level during the luteal phase of menstrual cycle? So definitely it will be progesterone, because a large amount of progesterone and a small amount of estrogen is produced by corpus luteum. So it's 1. Which of the following functions is performed by ciliated epithelium? Movement of food through intestine? No. Flow of renal fluid through PCT? No. Movement of particles through the bronchioles? Absolutely. Flow of blood through arteries? Not at all. So answer is 3. Very good, Mansa, Vrishti. Very nice. Now assertion. Oxytocin stimulate milk ejection from the mammary glands? Yes. Oxytocin stimulate the contraction of smooth muscles of myometrium. So oxytocin do cause the contraction of smooth muscle of myometrium, but again, both are correct but not the correct explanation of assertion. So answer should be B. Okay, or 2. Next. Which of the following hominid has a brain capacity of 900 cc and probably ate meat? So it was Homo erectus. So answer should be 2. Next. Metamorphosis. Metamorphosis will be seen in petromyzin or lamprey. Metagenesis is seen in obelia. Metamerism is seen in Nereis. Pneumatic bone in Cetacula. Okay, very good question, and the correct answer should be 1. Tegmina in a cockroach are. So these are mesothoracic wings. Okay, answer should be 2. Right. Which part of the human embryo possess the potential to give rise to all the tissues and organs of the body? Definitely inner cell mass. Okay, and not just the any other option that are given, trophoblast or endometrium or placenta. Easy. Assertion says inspiration is initiated by the contraction of diaphragm. So when diaphragm contract, okay, from dome shaped, it will become flattened. Okay, so that is right. The contraction of the diaphragm decreases the thoracic volume. No, it is wrong. So assertion is true, reason is false. Answer should be 3. Okay, answer is 3. Now read all the statements and figure out the correct statements. PTH stimulate the reabsorption of calcium by the renal tubule. So ultimately PTH, parathyroid hormone, increases the blood calcium level. So if it is stimulating the reabsorption of calcium from the renal tubules, definitely there will be increase in blood calcium level, which is right. Hyposecretion of insulin result in diabetes insipidus? No, it is related to ADH. Hypothyroidism during pregnancy causes exophthalmic goiter? No, it causes creatinism. Melatonin play a very important role in regulating the diurnal rhythm of our body? Yes. Thymosin promote the production of antibodies to provide humoral immunity? Yes. So we have A, D and E as the correct answer. So answer should be 3. Yes, very good Aditya, very nice, good. Next, which of the following decreases the blood pressure by causing vasodilation? So it is A and F that reduces the blood pressure, atrial natriuretic factor. Got it? The presence or absence of the hymen is not a reliable indicator of virginity or sexual experience? Yes. Line of NCRT asked in NEET. The hymen can also be broken by a sudden jaw or fall or jolt, insertion of vaginal tampon, active participation in some sports like horseback riding and cycling. Assertion is true, reason is true, reason is the correct explanation of assertion as well. Okay, so answer should be 1. Next, assertion says hint 2 generates blunt end fragments. Yes, this is right. And recognition sequence of hint 2 have 6 base pairs. Yes, both are true. So assertion is true, reason is true, but not the correct explanation of assertion. So answer should be 2. So it's good that even the options of assertion and reason are given in sequence, right? Green glands are the excretory organs in crustaceans, right? Prawns. So it is 4. Biopsy. It's a histopathological test. MRI. Okay. Now, MRI uses non-ionizing radiations. PCR is polymerase chain reaction, right? Will be matched with detection of genes that increases cancer susceptibility. And alpha interferons are biological response modifier. So A is matched with 3, B is matched with 4. Okay, so we got our answer. It should be 3. Very good, Mansa, Vrishti, very nice, Aditya. So it is 3. Now, the functional insulin has A and B chain linked to each other by hydrogen bond? No, they have disulfide bond. So this is wrong. Serum and urine analysis are effective tool for the early diagnosis? No, they will not help in early diagnosis. So both are incorrect. Okay, so answer should be 4. Yes, good. Okay, so with this we have completed the discussion of our zoology portion. Now it is time to discuss the physics portion. So are you all ready? The zoology portion was quite easy, isn't it? So thank you very much for attending and please wait for the physics discussion.
शूटिंग कैसे लगता है
वो निकलता है.
वो आ गई तो हाँ और क्या ये वाला चल रहा है पहले वो स्लाइड देख लो आप पीछे पीछे ये पीछे क्यों रहता है आगे ये आगे से आना चाहिए ना पीछे जाता है ये चलो ठीक है कोई बात नहीं क्या
पीछे आपका
नहीं ठीक है फर्स्ट है ना फर्स्ट क्वेश्चन यही है हाँ ये ठीक है ना चलो हाँ 91 के बाद ठीक है देख लेंगे चलो ठीक है चल सैम इसको सीधे कर दो थोड़ा आ रहा है So good evening to all of you. Let's start physics discussion, right? How are you all? Good evening, Vrishti. Chalo, let's start, right? So, what is the first question? Just a minute, let me change the pen once. Okay, fine. So, what is the first question? Let us see. Okay. If force, velocity and time are taken as fundamental units, then the dimension of mass are, right? So basically this question is based on, dimensions, right? So you know what is the dimension for force? Yes. So what is the dimension for force? The force is nothing but mass into acceleration, right? Right? So let us try to do it. So mass L t to the power minus 2, right? That is my force. So just a minute, I have the paper with myself. Chalo. So ml t to the power minus 2, that is my force. Velocity I can write. What is velocity? L t to the power minus 1, correct? And then we have t, correct? So from here we have to derive mass, right? So how we can derive mass? So we need to eliminate this L and t, right? So we'll divide force by velocity. So let us do that. Force divided by velocity we can do. So if we do that, you can see that we will get m and we will get t to the power minus 1, yes? And then we can multiply this by t, So if we multiply this by t, we will get f t by v is equal to m, right? Or I can say it is nothing but f t v to the power minus 1, correct? So that is my first option. Fairly very simple, right? Chalo, let's go to the next question. Car A is moving to the east with a speed of 10 kilometers per hour. Okay. So let us, I told you that, whenever you want to do a question in physics, try to visualize the question, right? So let us make a car, a small car will make. So let's say, this car is there and it is moving towards east with a velocity of how much? It is moving with velocity of 10 kilometers per hour, right? Okay. And a car B is moving to north. So let's say this is A and this is another car and this is B. It is moving towards north. And what is the velocity of this car? It is 10 root 3, right? Now they are saying that what is the velocity of car B with respect to car A? Yes. So they are asking you what is the velocity of B with respect to A, right? So whenever they have asked you with respect to, what do? You sit on one and try to analyze the motion of the other, right? Understood? So I'll sit on A. Let me sit on A, car A. So I'm sitting on car A. And what will I do? I will give this velocity in opposite direction to car B, right? So let's say this is my car B. It was moving with 10 root 3 and now it has another velocity in this direction opposite of this, which is 10, right? So my resultant would be somewhere here, correct? So I can fairly calculate, very simple, this theta I can calculate. What is tan theta? Tan theta would be 10 root 3 divided by 10, which is my root 3, which gives me theta as 60 degree, right? So you can see that this is basically north, this is west. So this is north of west, right? So 60 degree north of west, I have only one option. And you can also calculate the magnitude 20. How you can calculate? Root over this square plus this square, right? So that will give you 20, correct? Next question. Imagine a person standing on a weighing machine placed inside an elevator, right? So imagine that you are in an elevator, right? So let's say this is an elevator and you are standing here, right? Now placed inside an elevator. So there is a weighing machine. You are calculating your weight. The weighing machine is kept on the elevator and elevator is moving. The elevator first accelerates at constant rate A and then moves with a constant velocity and finally decelerates at the same constant rate A to stop, right? So what is happening? Basically, initially it is accelerating. So if let's say, this is not accelerating, what will happen? This weighing machine will weigh your weight as m into g, correct? Understood? But when the elevator is accelerating upwards, right? So let's say this is moving upwards with an acceleration of a. What will happen? We will have a pseudo force which is acting downwards if I sit on this frame. So basically then what will happen? One ma will also act here, and if it is moving with acceleration a, you will have m of g plus a, right? So your weight will increase basically, right? Understood? Your weight will increase. Now what they are saying, so they are saying that the maximum and minimum weights recorded are 800 and 640 Newton, right? So you tell me that when will the maximum weight come? When the elevator is moving upwards with an acceleration of a, right? So the total weight would be m times of g plus a. So they have given you actually the value of m of g plus a. So m of g plus a is nothing but 800, right? That is given to you. And minimum, when it will be minimum? When basically this is moving, this is decelerating, right? So if it is decelerating, it would be m times of g minus a, and that is given as 720, right? Sorry, 640, correct? Now there are two equations which you can solve. G value 10, you can divide both of them actually and m will cancel out. So g plus a upon g minus a, right? That would be 800 upon 640, and from here you can calculate the value of a, right? Putting g as 10, correct? So your answer will come out to be 720. The calculations I am leaving up to you, right? We are just doing the conceptual part, right? Understood? Clear? Okay. A stone falls freely under gravity. So let's do this. A stone falls freely under gravity. It covers distance d1 and d2 in the first 4 seconds and in the next 8 seconds. The ratio of d2 by d1 is, right? So again, try to visualize this question, what is happening. So, if I let's say from I'm here at the tower, right? And this is my ground and I'm dropping a ball from here. So let's say this is t is equal to 0, correct? So from here I have dropped the ball. What is my initial velocity? My initial velocity is 0 because it is dropped, correct? Now they are saying that it is dropped and it covers a distance d1 in first 4 seconds. So let's say in first 4 seconds it comes somewhere here, right? So let's say this is my t is equal to 4 seconds, correct? So it has covered a distance of d1, they are saying. Very good, correct? Now they are saying that in the next 8 seconds, it has covered a distance of d2, which means that it will fall further, and let's say it is here at t is equal to 12 seconds. Why I am writing 12? Because this difference should be 8. Another 8 seconds it has fallen, right? So 4 plus 8, that is 12. So this distance is d2, correct? Right? Now we need to calculate this ratio, correct? Okay. So what I'll do, I'll analyze both the motions from here itself, because my initial velocity is 0 there, and I can apply this equation s is equal to ut plus half at square, right? I can do that, right? So if you see, for this whole motion, the total distance covered is d1 plus d2, and the total time taken is 12 seconds, correct? 4 plus 8, which is 12, right? So I can apply these two equations. So I'll first apply for this part. Let's say this is A, this is B, this is C. So I'll first apply for AB, right? So if I write the equation for AB part, what will I write? I will write S, which is D1, is equal to UT, U is 0, half AT square, A is nothing but G, and T is nothing but 4 square, right? So this is my first equation, right? D1 is equal to half G into 4 square. The next is D1 plus D2. This is the equation for A and C, this motion, AC. So that would be again, U is 0 because I am starting from here. So U is 0, half G into total time. What is the total time? 12 seconds. So 12 square, right? Now from here, you have to calculate D1 and D2. Basically, you can subtract both of them and you will get the value of D2, right? D1 you can already calculate from here. You have to put G as 10, correct? So the calculations again, I am leaving up to you. This is the two equations, correct? Next, a block initially at rest slides down on a fixed rough inclined plane. So let's make a rough inclined plane here. So let's say this is my block and it is sliding down here, right? So this is rough. So let's say, the coefficient of friction is mu, correct? Let's say the coefficient of friction is mu, correct? Now, inclined at an angle of 45 degrees. So this is inclined at an angle of 45 degrees, okay, with the horizontal. Then the distance traveled by the block in the first 2 seconds is. So they have given you the time, which is the 2 seconds. So I know that the block is sliding for how much time? It is sliding for 2 seconds. Yeah, yes. Now I know that when it starts, it will start with an initial velocity of 0, correct? So initial velocity is 0. Can I find the acceleration here with which it is sliding? Yes, I can find the acceleration, right? Because, you see, from the mg part, g sine theta is acting here, right? G sine theta is acting here, and the frictional deceleration is mu g cos theta. This you might know, right? So the friction is acting above, right? So the net acceleration would be g sine theta minus mu g cos theta, right? So that is my net acceleration. Let me calculate this value. So you can calculate this value. So sine 45 and cos 45, they both are same. G and G you have to take 7 root 2. So let me just do it quickly. So 1 by root 2 G minus mu into G. So mu is how much? Mu is given as 0.3. So again you can calculate from here 1 by root 2 multiplied by 0.7 into G. G is nothing but 7 root 2. So this cancels out. This is 4.9 meter per second square. That is my acceleration, which is 4.9 meter per second square, right? So I have to take the, let us do it here. So now from here, how will you calculate? Distance is equal to ut plus half at square. Again the same, 4.9 into t square, which is 2 square. Now from here you can calculate the value, correct? Simple question. Let's move ahead. When a body performs uniform circular motion, no work is done during half a circular motion. In fact, in a circular, if it is moving in a circular motion, no work is done at all. Because why no work is done at all? Because at any point of time, the force, which is the centripetal force, and the displacement, they are always perpendicular, right? So in a circular motion, the centripetal and the acceleration, they are always perpendicular, and since work done is a dot product, it is 0, correct? It is always 0. Is that very, very clear? So force and displacement are always perpendicular to each other, and hence it is always 0. Simple. Let's move to the next part. A constant force of 5 Newton accelerates a stationary body of mass 500 gram through a displacement of 5 meter. Okay, fine. The average power delivered during the stages, right? So if you know what is the average power is given by F dot V, correct? So basically it is force into velocity, right? Fv cos theta. Now, so we have force which is 5 Newton. We have mass also, okay, fine. And we can also, we can also calculate for power as work done per unit time. So work done per unit time is also there, right? So from here you can also do like this, F dot x by t, where x is the displacement, because we have been given the displacement, right, which is 5 meter, correct? It is in the direction of force only. So we can write force. What is force? 5 Newton. What is displacement? 5 meter. Time, do we know the time? No, we don't know the time, right? So we'll have to calculate the time. So you can calculate the time by again the same equation, S is equal to ut plus half at square, right? So again, initial velocity is 0, so this is 0. Displacement is 5 meters, half. What is acceleration? Acceleration is force per unit mass. What is force? 5 newtons. What is mass? That is 500 grams, or I can say 0.5 kgs multiplied by t square, correct? So from here you can see that you will get t as 1, right? And you can put there and you'll get the answer as 25 watts. Clear? Next, ball P of mass m moving with velocity v collides with another ball q. So let's say this is my ball P and this is my ball Q, correct? And we have this P is of mass m, okay, and it is moving with velocity v, correct? Collides with another ball Q of mass 2m. So this is of mass 2m at rest. So this is at rest, okay. Final velocity of P just after the collision is. So this time the coefficient of restitution is given to you, that is 1 by 3, right? So the value of E is 1 by 3, right? So again, let's say it collides, and after the collision, P is moving with the velocity of v1 and Q is moving with velocity of v2. Let's say this is happening. This is mass m, this is mass 2m, right? So again, in any collision, the momentum is always conserved. So initial momentum would be equal to the final momentum. What is the initial momentum? M into v. That is my initial momentum. What is my final momentum? M times of v1 plus 2m times of v2. That is my final momentum, right? So from here you can see that you will get one equation, v is equal to v1 plus 2v2. That is my first equation, correct? Where will I get the second equation from? The coefficient of restitution is given to you, that is 1 by 3, right? So let me make the other equation also. 1 by 3 is equal to, so again, v2 minus v1 upon u1 minus u2, right? So this I am taking as 2. So v2 minus v1 upon u1. What is u1? v minus 0. So from here you can see that v by 3 is equal to v2 minus v1, right? So now you have two equations and two unknowns, and you have to solve for them, right? So you can calculate the value of v1 and v2 from here. Is that very, very clear? Next, a circular hole of radius 3 centimeter is cut out from a uniform circular disc of diameter 12 centimeter. So we have, a big disc. What is a disc? Disc is basically 12 centimeter in radius. 12 centimeter in diameter. So the radius is 6 centimeters, right? So the radius is 6 centimeters, and from here we are basically, taking a hole out from here like this, whose radius is 3 centimeters, right? So this is the case, correct? Clear? So basically in cavity questions, what do? We take the mass of the cavity, we take the mass of the remaining, and their total center of mass should lie at the same point, right? So this is the, before cutting out, this was the center of mass, correct? So what will I do? I will calculate the mass of cavity, correct? I will calculate the mass of the remaining part, and then both the combined center of mass should lie at the middle point, which is this. So let me say that this point is 0, 0. right? What is this point? This point would be 3, 0 because this distance is 3, right? And let's say the remaining part center of mass is somewhere here. I don't know what that is. Let's say that is x, correct? That is x. So first I need to calculate what is the mass of the cavity. So let's say the total mass of the disk is m, correct? So what would be the, surface mass density? That would be m by area. What is area? Pi r square, which is 12 square, right? This is my surface mass density. So what would be the mass of the cavity? Mass of the cavity would be m by pi into 12 square into the area of this cavity, which is pi into 3 square, right? So you can see that, sorry, just a minute. This is not 12, this is actually 6. So this is not 12, this is 6 actually. I'm so sorry. So the diameter is 12, radius is 6. Okay, so here also we'll have to do. So this is 6. So you will get m by 4. So this is a relation between the masses, correct? So now what you can do is, you can basically take the remaining mass. So what is the remaining mass? Remaining mass would be 3m by 4, right? So now the total center of the x coordinate of the center of mass should be 0, right? So let's say this is x. So 3m by 4 into x, m1 into x1 plus m2 into x2, that is m by 4 into x2. What is x2? 3 divided by total mass has to be 0. So from here you can calculate the value of x. You can see that it will come out to be, x will come out to be 1 from here, right? Clear? The moment of inertia of uniform circular disk. So we have a uniform circular disk of radius r and mass m about an axis touching the disk at its edge and normal to the plane of the disk, right? So basically it is like this. So let's say this is the axis, like this it is there. So we know about this axis. What is the moment of inertia about this axis? mr square by 2. And by using parallel axis theorem, you can calculate the value of I, right? Here, what will be the value of I? So that would be mr square by 2 plus mr square. And that is option number 4, right? So that is 3 mr square by 2. Clear? Earth shrinks to 1 by 8th of its original volume, right? So let's say this was the volume earlier. Now it has shrunk to how much? It has shrunk to VE by 8, 1 by 8th, right? So how would the radius have changed? So volume is given by 4 by 3 pi r cube, right? This is my volume. So now it has become 1 by 8 times of this, right? Which means that the radius would have become half. Then only it can become 1 by 8 because, that is r cube there. So which means that r has changed to r by 2. Yes, then only the volume can be half. Now, the duration of days, again, the angular momentum will be conserved, right? If, it has shrunk, there is no external force acting. So I can say that I1 omega 1 is equal to I2 omega 2, right? So now, because earlier I know what is the, omega 1. Time period is 24 hours of rotation. That is equal to 2 pi by omega 1, right? So from here I know what is omega 1, or at least I can replace this, right? So what is I of Earth? That is 2 by 5 mr square, right? Omega 1, that is equal to 2 by 5 m r2 square omega 2. So 2 by 5 you can cancel. So r1 square omega 1 is equal to r2 square omega 2. Using this relation, you know what is r1 and what is r2. This is r1 and r2 is basically r1 by 2. Yes or no? Correct? So this is r1 square. Omega 1 is nothing but this, 2 pi by 24. 24 hours, that is r2. What is r2? That is r1 by 2 whole square into omega 2. Omega 2 is 2 pi by t. You have to calculate t from here, right? So 2 pi gets cancelled. This r1 will get cancelled, and from here you can calculate, which will be 6 hours. You can see this is 4, 24 and 24 by 4 is 6. Yes. Next, a planet is revolving around the sun in which of the following. So this is a sun and a planet is revolving around this like this. The time taken in traveling DAB. So this is DAB, right? So how much area it is covering? This much area it is covering, right? So basically they are asking, so this is a application of Kepler's second law, that rate of change of area that is constant, correct? Understood? So you can see that the area subtended by DAB and area subtended by DCB, which is this one. So DCB is greater, right? Right. So since this area is greater, the time taken also has to be greater for the ratio to be constant, right? So time taken by DCB has to be greater than time taken by DAB. Is there any option like this? Time taken by DAB is less than BCD or DCB, whatever is it. So the first option is correct, right? Clear? Next question. What is the next question? A simple pendulum is placed inside a lift which is moving with some magnitude of a acceleration, right? So again, we have a lift like this, and let's say we have a pendulum like this, correct? So it is moving with some acceleration. Let's say that A is the acceleration, right? If the time period of the pendulum with the lift moving upwards and downwards are in ratio 1:2, right? So again, let's say it is moving upwards. You know that what is the time period of pendulum? 2 pi root over L upon effective acceleration, right? And if the elevator is moving upwards, the effective acceleration is G plus A, right? So in upward case, G effective is G plus A. So my time period in up is 2 pi root over L by G plus A, right? That is my time period in going up, right? What is the time period in going down? Again, it would be just negative of this. So that would be 2 pi root over L upon G minus A. Now the ratio of these two have been given to you, that is 1:2. So TU upon TD, that is given as 1:2. So you can see from here, let us try to calculate this. So let us try to take the ratio from here. So that would be root over G minus A upon G plus A, right? Is that clear? That TU, G plus A will come down. So from here you can, square both sides and then calculate the answer, right? Mathematics I am not doing. Again, the average depth of an oil well is 200 meter. So let's say this is my oil well and this depth is given to you, which is 200 meters. Okay, fine. If bulk modulus of oil is this much. So bulk modulus, what is bulk modulus? What is the formula for bulk modulus? Minus delta P, which is a change in pressure, minus delta P divided by change in rate of change of volume. So delta V by V, that is my bulk modulus, right? Okay. So this is given to you and the density of oil is given to you. So the density of oil is basically 1500, that is given to you kg per meter cube. Okay. The percentage compression at the bottom of the well is, so they are asking you what is the compression here which is happening, right? So again, compression means what they are asking you delta V, right? Basically they are asking you, they are asking you delta V by V into 100, percentage compression at the bottom they are asking you. You can calculate from here. So delta V divided by V is equal to minus delta P divided by B, that is my delta V by V, and I'll multiply by 100 both the sides to calculate the percentage. So that would be equal to minus delta P divided by B multiplied by 100, right? So what is delta P? Delta P is basically the pressure change, that is the rho GH, right? So what is the pressure here? What is the gauge pressure here? Rho GH, right? So rho GH divided by B. So minus is just showing you that it is a compression, right? So I'm ignoring minus for now. B, what is B here? B is 8 into 10 to the power 8 into 100. Can you just put the values of rho G and H and calculate the answer? Fairly simple, right? So let us move forward. Liquid A rises to a height of 10 centimeters, okay, in a capillary tube, and a liquid B falls to a depth. So it is falling to a depth of 2 centimeter in the same tube, okay. The density of A and B are this and this respectively. The contact angle of A and B with the tube is 0 and 135 in both the cases. So there are two cases basically, right? So liquid A and liquid B, right? So let us see what is happening here. So it is rising to a height of 10 centimeters, right? And this is falling. So for denoting a fall, I am writing a minus sign. So minus 2 centimeters. The density of A and B, so density is also given of the liquids, which is 1 gram per centimeter cube, right? And density here is given to you as 10 gram per centimeter cube. The contact angle is given. What is theta given to you? Here it is 0, here it is 135 degrees, right? So basically you have to calculate the surface tension ratio. This is what you have to calculate. So how will you calculate? There is a standard formula. H is equal to 2t cos theta by rho GA. So the height of rise is given by 2t. In this case, it would be the surface tension, which is S cos theta divided by rho GA, right? So for first you have to write. So for I am writing for case A. So for case A, it would be 10 is equal to 2 times of the surface tension of A multiplied by cos 0, which is 1, divided by rho, which is 1, into G into A. So A is basically the radius of the tube, which is constant in both the cases. Again, in the second I will write minus 2 is equal to 2 into surface tension of B multiplied by cos 135. What is cos 135? Minus 1 by root 2, right? So that is minus 1 by root 2. Ignore it. Density is 10 into G into A. Now you have to just take the ratio of both of these equations, right? And you will get the value of SB by SA. Is that very, very clear? Next. A steel wire of length 20 centimeter and area of cross section 1 mm square is tied rigidly, right? At both ends. So basically this is a steel wire, let's say. Okay. So the length is 20 centimeters. Correct. Area of cross section is given to you. What is the area of cross section? That is 1 mm square. That is my area of cross section. Temperature is 40 degrees Celsius, fine. When the temperature of the wire is changed from 40 to 20, right? So it is being contracted, right? So when it goes from 40 to 20, it will receive a contraction. But since it is fixed, it will not allow it to contract, which means that actually, you know, at the final stage, it is in the expanded state, right? Okay. So delta T I know, which is the magnitude of delta T is 20, right? Tension developed, you have to calculate. Why will the tension develop? Because of this, you know, strain. Because you know, these both ends, they are not letting it to contract. So there would be a tension in the string. Okay. So again, Young's modulus is given by force per unit area divided by delta L by L, which is the strain. So basically this is a thermal strain which is coming here. So this is a thermal strain, right? This is a thermal strain due to the change in temperature, correct? So F is nothing but the tension which is developed. So I can calculate F from here as gamma into delta L by L. This will multiply here, multiply by A, right? So tension will be given by gamma is given to you, Young's modulus is given to you, 2 into 10 to the power 11 multiplied by delta L by L. What is delta L? Delta L is given by L alpha delta T, right? So delta L by L would be nothing but alpha delta T. So alpha delta T, we'll put alpha delta T and into area. So now you have to just put the values. Alpha is given to you 1.1 into 10 to the power minus 5, delta T is 20, and area is also given to you, which is 1 mm square. And do put all of these in SI units. So 1 mm square would be nothing but 10 to the power minus 6 meter square, right? So you have to put that into meter square, right? So please remember the equations. I mean, the values that you have to put. I'm just writing it here just for the sake of mentioning it, right? Delta T is 20 and the value of alpha is already given to you, that is 1.1 into 10 to the power minus 5, right? So from here you will calculate the value, correct? Next, a composite slab is prepared with two different materials, right, which is A and B of same cross section and connected in series. Okay, so we are connecting two here like this. Okay. Fine. Cross section is same. They are connected in series. The relation between the coefficient of thermal conductivities of these lengths are given as Ka is equal to Kb by 2. So let's say this is Ka and this is KB. So there is a relation which is given to you, Ka is equal to Kb by 2. Okay, fine. Length is twice. So if this is LA and this is LB, we have the ratio LA is equal to twice of LB. This also we know. If the temperature of the outer faces are 75 and 50, so let's say this is end A, so this is 75 degrees Celsius and this is 50 degrees Celsius, correct? Then the temperature of the common surfaces, so this you have to calculate, which is T, right? So how will I calculate this T? Basically, what is the concept? Since both of these are connected in series, the heat current will be same, right? So first I will apply the conduction equation between this point and this point. So what is that? So rate of transfer of heat, which is dq by dt, that is change in temperature T minus 75 divided by L by Ka. So LA divided by K of this. So I am writing it in numerator, K into area, right? And again, I will again apply this equation from, either you can apply from 75 till 50 or you can also apply from T to 50. So I'll apply from T to 50. So that would be 50 minus T divided by LB multiplied by KB into A, right? So now you have to equate both of them. That you can do. So I'm just writing it directly, T minus 75 divided by LA into Ka, that would be equal to 50 minus T divided by LB into KB. Now you have the relations, you just put the relations, cancel from both the sides and calculate the value of T, right? I think that comes out to be 55. That is just pure calculation. Clear? Next question. Work done on compressing 1 mole of monoatomic gas adiabatically. So we have 1 mole of gas. We are basically, compressing it. That is a monoatomic gas. So gamma is basically 5 by 3, right? Adiabatic compression is happening at through a change in temperature of 20 degrees Celsius. So delta T is given to you, which is 20 degrees Celsius. Work done is W. So what is work done? Work done is NR delta T divided by 1 minus gamma. That is the work done in adiabatic process, right? So W is equal to N, which is 1, R delta T is 20 divided by 1 minus 5 by 3. That is my W. Again, the work done on compressing 6 moles of rigid diatomic gas, right? So work done for diatomic gas would be again the same adiabatic. It is a process is same adiabatic. So again, just the number of moles will change, 6 into R into to the same change in temperature, which is 20 divided by 1 minus gamma. What is gamma for diatomic gas? Yes, what is gamma for diatomic gas? That is 7 by 5, right? So basically you will put 7 by 5 here, and now you can take the ratio of both of them and calculate the value of WD in terms of W, right? Understood? Clear? So that will come out to be 10 watts. Mathematics you have to do. I'm just explaining you the concepts, correct? Okay. Next. Given below are 2 statements. One is assertion. So let's see this assertion reason. Adiabatic expansion leads to heating effect, right? So which question is this? 109. Okay. So now you have to tell me. This is again a very simple question, right? Adiabatic expansion leads to heating effect. Is that true? So from first law of thermodynamics, you know that delta U is equal to delta Q plus W, right? In adiabatic process, there is no heat exchange, correct? There is no heat exchange. So delta U, sorry, this is just a minute. This is not plus, this is minus. Right? Now this is 0. So minus delta U is delta W, or change in work done, whatever you want to say, right? So if the expansion is happening, right? Just a minute. If the expansion is happening, there can be a cooling effect also, right? There can be a cooling effect also. So this statement is basically wrong, correct? The next is, in adiabatic expansion, gas expands at the cost of internal energy. Yes, that is true, because there is no heat exchange, right? There is no heat exchange. So that is true. So first is false, second is correct. So that gives you 4, right? Let's move to the next question. Two blocks of ice when pressed together join to form one block because. So when you press both of these blocks, what will happen? There is a pressure which is acting here, and because of the pressure the melting point decreases, and because of it melts, and as soon as you remove the pressure, basically it again solidifies and it welds. So basically what is happening, so of heat is produced during pressing, okay. Melting point of ice decreases, that is correct, with the increase of pressure, right? Understood? So basically melting point decreases, and because of which, you know, there is a melting point of ice decreases with increase of pressure. That is correct, right? Understood? Next. A cylinder consists of a gas mixture of helium and oxygen. So there is a gas mixture, helium and oxygen. Sorry. So I made hydrogen. I'm so sorry. Yeah. So that is helium, which is a monoatomic gas, and we have a oxygen which is diatomic. So we have monoatomic and diatomic gas. It is a mixture. We have to calculate the ratio of specific heats of the mixture, right? So what is the formula? Gamma of mixture is given by Cp of mixture divided by Cv of mixture, right? And you know how to calculate Cp of mixture, right? So what is Cp of mixture? Cp of mixture is given by, let's say this is 1, this is 2. So N1 Cp1 plus N2 Cp2 upon N1 plus N2, right? And what is Cv of mixture? Cv of mixture is N1 Cv1 plus N2 Cv2 divided by N1 plus N2, correct? What is N1? So we have 4 grams of helium. Again, the atomic mass is also 4. So N1 is basically, what is N1? N1 is basically 1. And what is N2? N2 is also 1 because for oxygen it is 32 grams, right? So N2 is also 1, correct? So N1 is 1, N2 is 1. Okay, and can we also write Cp1? So what is Cp1? That is a monoatomic gas. And what is Cv1? Right? So this, you know how to write. What is Cv of a monoatomic gas? That is 3R by 2, right? So that is 3R by 2. And this is 5R by 2, right? What is Cv of diatomic gas? 5R by 2, right? And 7R by 2. Now you have to put all of these values in these formulas and calculate Cp and Cv and then calculate the ratio, right? That is pure mathematics. So you'll get the answer as 3 by 2, correct? Let's move ahead. For a molecule of an ideal gas, the number density is 2 root 2 by 2 root 2 into 10 to the power 8. So small n is given to you, that is 2 root 2, which is the number density, into 10 to the power 8. And the mean free path is, so lambda value is given to you, which is the mean free path, that is 10 to the power minus 2 divided by pi. The diameter of the gas molecule is, okay. So you know the formula for mean free path, right? 1 by root 2 pi n d square, where d is the diameter, right? Whereas d is the diameter. So what is d square from here? D square would be 1 by root 2 pi n lambda. You have to just put the values. You already have all the values. You have value of n, you have value of lambda, you have root 2. You just have to put the values and take the under root. Clear? That is very simple maths. Yes? Let's go to 113th question, right? So what is the next question? The next question is a cylindrical tube open at both ends. So we have a cylindrical tube. Let's make a cylindrical tube which is open at both the ends. So this is open at both the ends, okay. Has a fundamental frequency f in air, right? So if it is open at both of the ends, at the ends we'll have a antinode, right? So it will be like this. For fundamental frequency it would be like this. Let's say this length is L, right? So in terms of lambda, can I write the length L, right? This is lambda by 4, lambda by 4, which is lambda by 2. So lambda by 2 is equal to L, lambda is equal to 2L, right? And if I want to write the frequency, how will I write? So frequency is given, that is F is equal to velocity divided by lambda, which is 2L, right? That is my first equation. Now they are saying that the tube is dipped vertically in water. So now we are dipping this tube vertically into water. Let us dip this tube vertically into water, right? So let's say, I have dipped this in water till here, right? So that 60% of the tube is in water. 60% means this length, out of this total length, 0.6 L is here and rest 0.4 L is outside, right? This is what it means, correct? Now, then the fundamental frequency of the air column is. So what will be the fundamental frequency here? It will be like this, because now this end will act as, this is a closed end now. So this will act as a closed end. So now you can see that in how I can write lambda. Lambda by 4 would be equal to 0.4 L. So lambda would be equal to 1.6 times of L, right? So now what is the fundamental frequency here? Fundamental frequency would be equal to my velocity, which is V, divided by 1.6 L, right? And can I write V from there? Right? I can write V from there, which is F into 2L divided by 1.6 L. So this is cancels out. You can calculate from here what it would be. 5 by 4, I think, right? So that would be 5 by 4. Clear? That is 5F by 4. Is that very, very clear? Can we move on to the next question, right? Any doubts? Okay, let's go ahead. Two vibrating strings A and B produces beats of frequency 8 Hz, right? So we have two vibrating strings A and B. They are producing beats of 8 Hz. So what is beat basically? It is a difference in frequencies, right? It is a difference in frequencies, that is 8. But we don't know which one is higher, right? Which one is higher, we don't know. That is what we have to identify here. So that's why they have given you the second information. What is the second information? The beat frequency is found to reduce to 4 Hz if the tension in the string B is slightly reduced. And now we know that frequency is basically directly proportional to root of tension, if you remember, right? So if you are reducing the tension, actually you are reducing the frequency, right? So if you are reducing the frequency, and if the if by reducing the frequency, the beat frequency has reduced, which means that we are reducing this part, correct? So it has to be FB minus FA has to be equal to 8, because if I reduce the tension, this component will decrease and this difference will decrease, and that will give me 4, right? Which means that FB minus FA is 8. Now they have given you the initial frequency of A, which is 320. Now you can put 320 here. That would be equal to 8, and FB would be equal to 328. Clear? That is clear, right? Next. A ray of light is incident at an angle of 60 degree on one face of a prism. So let me make a prism here, right? So we are in at an angle of 60 degree on one face of a prism of angle 30 degree. So the angle of prism is 30 degree. We are incidenting a light here like this at an angle of 60 degrees, correct? The ray emerging out of the prism makes an angle of 30 degree with the incident ray. So this is my incident ray, correct? This is my incident ray. With the incident ray, the refractive index of the prism. So you have to calculate the refractive index of the prism. Okay. So let me do one thing. So they are saying that this is making an angle of 30 degree. Okay. At an angle of 60 degree and on one face of the prism of angle 30 degree, the ray emerging out of the prism makes an angle of 30 degree with the incident ray. So basically the ray which is emerging, it is something like this. Just let me make a diagram first. So let's say it is going something here like this. Right? So this is the thing. And if I make it back here, this angle they are saying that angle is 30 degree. Right? I think this is what they are saying. Let me see here also. Right. So this is what they are saying, right? So this angle has to be 30 degree. This is what they are saying. Okay. Fine. The refractive index of the prisms, right? So basically they have given you the deviation. What is this? This is a deviation. This is a deviation. And I know that what is deviation? Deviation is I plus E minus A. Correct? That is my deviation. Correct? So deviation is given as 30 degrees. That is equal to I. I is 60. E minus A. What is A? A is 30. So from here, can I calculate the angle of emergence? So you can see that angle of emergence will come out to be 0. Angle of emergence will come out to be 0, which means that the ray is incident perpendicularly on the other part, right? So I'm redrawing this diagram once again, and I'll show you how this diagram should be actually drawn. So let's say this is the incident ray, right? So it is going something like this. I mean, just perpendicular it is going and going like this, right? So E is 0 basically, right? So this is going like this. This is the thing which is happening. So now this refraction angle R2 is also 0. This is 30, which means that this refractive angle is nothing but 30 degrees. This is 60. Now you apply Snell's law here, guys. So how will I apply Snell's law? Sine 60 is equal to mu times of sine 30, right? So you'll get mu as root 3. Clear? You'll get mu as root 3, right? Next, a plano-convex lens is made up of refractive index 1.6. So we have a plano-convex lens, okay? So we have a plano-convex lens like this. Refractive index is given. So what is the refractive index? Refractive index is 1.6. Radius of curvature is given to you. What is the radius of curvature? That is 60 centimeters. Okay. The focal length of the lens is very, very simple question, right? You have to just use the formula. What is the formula? 1 by f is equal to mu minus 1 upon r1 minus 1 upon r2, right? So what is r1 here? R1 is infinite because it is plane surface. What is R2? R2 is minus 60 actually, right? Because we are going in this side and radius is in opposite side, right? So you just apply the formula. 1 by f is equal to 1.6 minus 1. 1 by infinity is 0 minus 1 upon minus 60, right? So you can calculate this value by yourself. Is that very, very clear? Next. A Young's double slit experiment setup is immersed in water with a refractive index of 1.33. Okay. It has slit separation is given. So slit separation, which is small d, is given to you, that is 1 mm, or I can say 10 to the power minus 3 meters. The distance between the slits and the screen, which is capital D, is given to you as 1.33 meters. Okay. If the incident light has a wavelength of 6300 angstroms, okay, in vacuum. So this is in vacuum. Please mind, this is in vacuum, not in water, right? So you might do this wrong by taking this in water, right? So we have to calculate the fringe width. What is the formula for fringe width? That is d lambda in medium divided by small d, right? So what is lambda in medium? So lambda in water would be equal to lambda in vacuum divided by mu, right? So which is 6300 divided by mu, that is I think 1.33, right? So 1.33. So you calculate this value of lambda here and then you put it there, right? So beta would be equal to capital D, which is 1.33 multiplied by lambda, which is 6300 divided by 1.33 upon small d. What is small d? 10 to the power minus 3, right? So from here you can calculate the value. Is that very, very clear, right? Next. Magnifying power of an astronomical telescope for normal adjustment is 10. So again, f0 by fe is given to you. What is that? 10. The length of the telescope, which is f0 plus fe, length of the telescope f0 plus fe is given to you as 110. From here you have to calculate f0 and fe. When the image is formed in the near point, so if the image is formed at the near point, right? So they are asking you, when the image is formed at the near point, what is the, just a minute, magnifying power of the telescope when the image is formed at the near point and the object is at infinity. Okay, so what is the formula in such case? That is f0 by fe, right? f0 by fe 1 plus fe by d. That is my formula, right? So you calculate f0 and fe from here and you put that into this formula and you will get the answer, right? Is that clear? Next. The electric flux from a cube of edge L is phi. When a charge, so we have a cube here like this, right? So we have a cube. Electric flux from a cube of edge length L is phi when a charge is enclosed in it. So there is some charge here which is enclosed in it. So what is the flux? Flux would be equal to q upon epsilon naught because the charge enclosed is q. If the edge of the cube is made of length 2L by 3, right? So if it is made of 2L by 3, then the electric flux from the cube is, again, that would be same phi, right? Because q is kept inside, whether the dimension is L or 2L, the flux does not depend on the dimension, right? Next, in the following circuit, the equivalent capacitance between the terminal A and terminal B is, so we have, so again, this is a Wheatstone bridge, right? So this will cancel out. 2 by 2 is equal to 2 by 2, so this will cancel out. So these two are in series. So if they both are in series, what will be the equivalent of this? That would be 1 microfarad, right? What would be the equivalent of this? That would be again 1 microfarad, and both of these are in parallel, so total will become 2 microfarad, right? Clear? So that would be 2 microfarad. Yes? Very good, Ria. 2 microfarad. Next. Clear? In a meter bridge experiment, the balance point from left is 37.5. So let me make the meter bridge first. So from left hand side, the balance point is how much? The balance point is 37.5. So what would be this? This would be, I think, 62.5, right? Because 100 minus 37.5, that is 62.5, right? If the right gap has a conductance of 0.1, so this is a right gap, conductance is given, not the resistance, right? Resistance would be 1 by conductance, so that would be 10. Let's say the resistances are, then the left gap resistance is. So can you do that? R by 10 is equal to 37.5 divided by 62.5. Fairly simple, right? So you can calculate from here. I think what would be the answer? So answer would be 6, right? So you just calculate, the answer would be 6, correct? Next. Is that clear? Consider a wire of length L which has a resistance of 5 ohms. So we have a wire whose length is L, resistance is 5 ohms. Okay, fine. Applying an external force, the wire is elongated such that its length becomes 3L. So now we have applied an external force and this length has become 3L. Assuming the resistivity and density of the material is unchanged, the resistance of the wire changes by, right? So again, you know what is resistance, right? So how do we calculate resistance? Rho L by A, right? So here what else is changing apart from length? Area is also changing, right? Because the volume has to remain constant, right? So let's say the area here is A1 and area here is A2. So A1 into length should be equal to A2 into the next length, which is 3L. So A1 has to be equal to 3 times of A2, correct? So now it is just the ratio basically that you have to calculate. The resistance in second case would be rho, which is constant, length is 3L and area is A2, right? So A2 you can calculate A1 by 3. So that would be rho into 3 times of L divided by A1 by 3. So that would be equal to 9 times of rho L by A1. And what is rho L by A1? Rho L by A1 is nothing but the first resistance, which is given to you as 5. So that would be 9 into 5, which is 45, correct? Very good. That would be 45. A short bar magnet placed with its axis at 30 degree with an external field of 800 G. So first let us convert this magnetic field into Tesla. 800 into 10 to the power minus 4 Tesla, correct? Experiences a torque of this much. So torque is given to you. What is torque? 0.016. Okay. So what is the torque experienced by the magnet, by the way? That is M cross B. Or I can say it is nothing but MB sine theta. Right? That is my torque. Theta is given to you as 30. So sine 30 is half. Torque is 0.016. They have asked you what is the magnetic moment. So M we have to calculate. B you already know, 800 into 10 to the power minus 4 and sine theta, that is half. Just apply the maths and calculate the answer, right? Fairly very simple, right? Understood? Next. A particle of mass m and charge q moving along with speed v in a circular path of radius r, okay, has a magnetic moment mu. So basically we have a charge, let's say that is mass is m, it is moving in a circle of radius r, right? The magnetic moment is mu, okay. Well, how do I calculate the magnetic moment? Magnetic moment is nothing but I into A, right? And how I can calculate the current? Current is nothing but charge per unit time. So charge is Q, time is 2 pi r divided by velocity, which is v, multiplied by a, right? So that is my thing. Now they are saying, what is they are saying? They are saying that if the mass of the particle is doubled, okay, and maintains the same speed, so v is same while revolving in the circular, then the magnetic moment will. So you can see that magnetic moment is not dependent on the mass. Can you see? It is not dependent on the mass, right? A is nothing but pi r square, so it is not dependent on the mass, so it will remain unchanged, right? Because it is not depending on the mass. Clear? Hello Ashish, you are very late. We are already ahead. Yes. Understood? Any doubts? Chalo, let's move ahead. Okay. Now, this is a good question to make you do it wrong, right? Because we have a habit of memorizing this formula that EMF is minus d phi by dt, right? Yeah, it's okay. You can see the recording, right, Ashish? Okay. Magnetic flux, let's move ahead. Magnetic flux linked with a coil is given by phi is equal to 3t square plus 2t plus 4, right? Then the average EMF induced in the first 2 seconds is, right? So please note on this word that it is asking you the average and not the instantaneous, right? So if it would have asked you instantaneous, then you would have done d phi by dt and you have put t is equal to 2. But here you will not do that because they have asked you the average. So how do I calculate the average EMF? I calculate the flux at 2. I calculate the flux initially and then I divide by the total time, which is 2, right? Understood? So fairly simple. You can calculate what is flux at 2. That would be 3 into 2 square plus 2 into 2 plus 4. That is my flux at 2. And what is flux initially? That is 4, right? So you can just calculate these values, take the difference and then divide it by 2, right? Understood? That will give you, I think, 8 volts. Just do the calculation. I think I'm not sure what is the calculation. 4 into 3, 12, 12 plus 4, 12 plus 4, 16, 16 plus 4, 20, 20 minus 4, 20 minus 4, that is 16, 16 by 2, that is 8, yes. So that will come out to be 8, right? So Riya, it's not 1, it's 8 volts, right? It's not 14. Please check the calculation, correct? Okay, next part. The next is a long solenoid of radius 1 mm has 100 turns per mm. Okay, so number of turns per unit length is given to you. What is that number of turns per unit length? 100, but do remember that it is per mm, right? So don't make a mistake, that is turns per mm. So the length is mm, not meters. That is what I'm trying to tell you. The radius is 1 mm. Okay, if 1 ampere of current is there, so 1 ampere of current is there, flows in the solenoid, the magnetic field strength at the point on the axis of the solenoid is. So what is the magnetic field inside the solenoid? It is given by mu naught Ni, right? Clear? Mu naught Ni. So mu, what is mu? 4 pi into 10 to the power minus 7. What is N? 100 divided by, it is 1 mm, right? Per mm. But I have to calculate it in meters. So 10 to the power minus 3 multiplied by current, which is 1 ampere, right? So from here you can see the value will come out to be, so pi you take it as 3.14, right? So I think it would be 12.56, 3 plus 2, 5 minus 2. So that would be minus 2 here, right? Clear? Understood? Okay, next. Two concerned coplanar circular conducting loops have radii r. So one is of capital R and another is of small r, right? These are two coils. So this is very, very important that they have given you that, the capital R is greater than small r. If they would not have given you, it would have been a very difficult problem to solve. Their mutual inductance you have to calculate is proportional to, okay? So what is mutual inductance? It is basically the change in flux which is happening due to the current in the change in the other coil, right? So let's say the current here is I, and because of this change in I, because of this I, how much is the flux changing in this coil that we have to calculate, right? So flux would be B, the magnetic field here into area of this, which is pi r square, is equal to mutual inductance into the current, which is I, right? And what is B here? B is nothing but mu naught I divided by 2 pi, sorry, not 2 pi, mu naught I by 2r. Mu naught I by 2 into capital R into pi r square, that is equal to m into I. So you can see that it will be directly proportional to small r square by capital R. Is that very, very clear? Right? Small r square by capital R. Right? Clear? Yes. That is C option. No, Ashish, D is wrong. It is not capital R square by small r, it is small r square by capital R, right? Because we are calculating the flux change here, right? Understood? A current of 6 ampere is flowing at 220 volts in the primary coil of a transformer. Okay, if the voltage produced in the secondary coil is 110 volts and 40% of the power is lost. Okay, so what is the initial power? Initial power is V, which is 220 multiplied by 6 ampere. That is my initial power. What would be my output power? That would be, since 40% is lost, what do I am getting? I am getting 60% of this, right? So that is 0.6 multiplied by 220 into 6. That is my output power, right? Clear? Right? This is my output power. And what is output power? Output power would be voltage, output voltage multiplied by output current. What is output current? I don't know. That is what I have to calculate. But I know this V, which is 110, right? So that would be 0.6 multiplied by 220 multiplied by 6 is equal to 1111. Where is this? Where is the output voltage? Yeah, this is 1100 multiplied by current. Can you calculate the current from here, right? Yes? Yeah, you are correct, Ashish. That is 0.72. Is that very, very clear? Chalo. Let's go to the next question. At an instant, a plane electromagnetic wave has its magnetic field in the direction of the vector I minus j, right? So magnetic field direction is given to you, that is I minus j, right? And its electric field is in the direction I plus j. So electric field ka direction it is given to you, right? That is I plus j, है ना? अभी हमसे क्या पूछा? The wave is traveling along which direction? So it is very, very simple. That is E cross B, right? So you have to take the cross product. Cross product you can take, and when you take the cross product, you will get minus k cap here, right? So basically you will get minus 2k cap, right? And that would be the minus z direction. I hope you know how to take the cross product, right? That I am not telling you. Do it in basics of, vectors. You might have learned. Clear? The de Broglie wavelength of an electron having kinetic energy 100 electron volt is. So we have to calculate lambda. What is de Broglie wavelength? De Broglie wavelength is h by p. P is basically the momentum, right? And momentum you can write in terms of kinetic energy. That is root over 2m into kinetic energy, right? Now you just have to put the values. These are just calculation wala part, right? So 6.6 into 10 to the power minus 34, that is my h. What is mass of an electron? Mass of an electron is 9.1 into 10 to the power minus 31, right? And what is kinetic energy? That is given to you. I think that is given to you, which is 100 electron volt. Don't forget to convert it into joules, right? So 1.6 into 10 to the power minus 19, right? So that is my values. You have to just put it there, calculate the value, and I think it would be around 122 it should come. Okay, so nearest answer to 122 would be 124, right? Understood? Do these type of easy questions come in NEET? Yes, obviously they come. Why won't they come, right? They want you to score, right? They want you to score. If it is easy, that's good that your preparation is on track. If you are finding it easy. Okay. Right? Very good, Ashish, you are right. I hope you get full in coming exam. Okay, let's move ahead. A beam of photons with an energy of 10.5 electron volts strike a metal plate. Okay, so this is the energy with which it is striking, which is 10.5 electron volt. The photoelectrons are emitted with maximum velocity of 1.6 into 10 power 6 meter per second. So what is Vmax here? Vmax is given to you as 1.6 into 10 to the power 6. The work function of the metal is, so we have to calculate the value of phi, which is my work function. Okay, so again it is simple, right? Okay, so let us calculate. So this is basically based on photoelectric equation, which is kinetic energy is equal to the energy which is incident minus the threshold frequency, right? Clear? So now it is very simple. What is kinetic energy? Half mv square, half into 9.1 into 10 to the power minus 31 into velocity, which is given to you 1.6 into 10 to the power 6 whole square, right? But please remember that this will come in joules, right? This will come in joules, but you know this energy is given in electron volts. So you have to divide this by 1.6 into 10 to the power minus 19 to get this thing in electron volts, right? So this is equal to 10.5 minus 5. Now you have to do the calculation part, right? Clear? So these are some of the very easy questions, like the questions on electromagnetic, the photoelectric effect. Please do it very religiously, right? Okay. The ratio of maximum to minimum wavelengths in Balmer series of an hydrogen atom is, right? So again we'll use this formula, 1 by lambda is equal to Rz square 1 upon n1 square minus 1 upon n2 square, right? So that is a formula. So when will the maximum come? When will the maximum come? So whenever is the maximum energy emitted, right, then the lambda would be minimum, right? So maximum energy is emitted when it goes from n is equal to, n1 is equal to 2 to infinite. That is where the maximum energy is emitted. So the wavelength would be minimum. So in this case, the wavelength would be minimum, right? And when it is maximum, when it goes from 2 to 3, here it would be maximum. So you just have to put the values here and then take the ratio, right? Clear? Easy peasy, right? Okay. So I'm not doing the calculations. You can do the calculation by yourself, right? Yeah, that is correct. Nearest orbit, maximum length. The nearest jump would be the maximum wavelength, right? So n2 2 is equal to 3 when it will jump, it will give you the maximum wavelength, right? If 5 milligram of U-235 is undergoing fission with the slow neutron producing barium and krypton. So basically uranium U-35 is decaying into barium and krypton, right? This is a fusion reaction and they are saying that the approximate energy released is, so they are asking you what is the total energy released. If given that the energy released per fission is 200 mega electron volt. So in one, if one atom is getting going into this fission, it is giving you 200 electron volts, right? But there is no one atom here, right? It is 5 milligram of uranium. So which means that we have to calculate how many atoms of uranium 235 are there, right? So how do we calculate? By using the mole concept, right? So weight, which is 5 into 10 to the power minus 3 divided by the molar mass, which is 235, multiplied by the Avogadro number, right? That will give you the number of atoms, right? Understood? Clear? And if in one atom 200 electron volt is emitted, in n atoms how much it would be emitted? N into 200. Clear? So that you do. Understood the concept? Which of the following represents the forward bias diode? Again, what is forward bias? So I'll explain you what is forward bias. When this is connected to higher potential, this is lower potential, then this is called the forward bias, right? There is no current here. What you can see, this is minus 3 volts, this is plus 2, this is plus 5, this is minus 2, right? So again, in this is greater, so this is not the answer. Again, this is greater, this is not the answer. Again, this is greater, this is not the answer. In this, 0 is greater, so this is the answer, right? So that is forward bias. Yes? Clear? Understood? So this question came in CT, yeah. The output of the given logic gate is similar to the output of, so again, you can do it by Boolean also. If you don't know the Boolean, you can try the values also, whatever you feel like doing, you can do. I'll try to do it by Boolean, right? So this is basically a NOT gate, right? So if A is here, you will get A bar here. If B is here, you will get B bar here, right? And this is basically a NOR, right? So what you will get? You will get A bar plus B bar whole bar, and that is nothing but A into B. So this is a AND gate basically, right? Clear? Right? So I think we are done with this. I hope this is clear, right? Best of luck for your exams, and I hope that you do well. Bye-bye, take care, and a very good night.
Hello students, welcome to one more amazing session from Infinity Learn. This is the last paper to be discussed for the chemistry. This is Dr. Suresh, Faculty of Chemistry for you. So how you are doing? So how is your just? So last countdown hours for your NEET examination. Infinity Learn is always for you to support you guys. Okay. So let's start this GT paper NEET grand test आज के लिए. So quickly we'll do it. 2N2O5 gives rise 4NO2 plus O2. The rate of decomposition of N2O5 to the rate of formation of O2 is for disappearance of N2O5 and the formation of oxygen. It is for one molecule appearance, two molecule has to disappear. It should be double. Is it right? Okay, children, one more question. Hi, Manasa. Good afternoon. Please confirm is my voice is audible to you all? Is my voice is audible to you all? Yeah, it is double. Okay. So let's see one more. In the qualitative analysis, the identification of cation and anion, a salt given by group 5. Group 5 are going to be calcium and barium. They are precipitated as carbonates. They are precipitated as carbonates. Okay. Chlorides, sulfides, hydroxides and carbonates. Okay, chlorides, sulfides, hydroxides, sulfides again in the basic medium, then carbonates. Okay, that is the order of the cation group separation. The molecular conductance of barium 2 plus and Cl minus are. So barium chloride is given as ionic mobility of barium 2 plus and ionic mobility of Cl minus into 2. So these values are given. It is given as Cl minus, it is 127 plus 2 into 76. So this is your molar conductance. And it is asked about equivalent conductance, that is going to be molar conductance upon 2 because its valency is going to be 2. Hope you understand. And the answer is going to be 139.5. And talking about which of the following is an example of pseudo first order reaction, that is ester hydrolysis is a pseudo first order reaction, my dear students. This is an ester hydrolyzed, it is a pseudo first order reaction. Ria and Mansa, yeah, it is going to be 4. That's fantastic. And also you can see the vapor pressure of water, it is going to be 30 if the mole fraction of the water is 0.9 and the vapor pressure of the solution is going to be how much? P0 minus Ps upon P0 is equal to mole fraction of solute. Mind it. Here it is a trick. Mole fraction of A plus mole fraction of B is equal to 1. So that I just want to say P0 is equal to 30 minus Ps upon 30 should be equal to 1 minus 0.9 is going to be 0.1. You can solve it for Ps so that you're going to get 27 mmHg. So got it clear? That's good. Fantastic. Fantastic. Now the osmotic pressure, so pi is equal to sucrose I value is equal to 1. Okay, CRT. C stands for molarity, weight by molecular weight, 0.0821 into temperature is going to be 17 degrees centigrade, 290 Kelvin. So weight is given how much? 1.75 gram. Sucrose 340 2 into 0.0821 into 290. So that is going to be the answer. So answer 3 is the correct answer. Just check how these temperature values are changed. Only use Kelvin square to solve this question. Okay, only Kelvin square. Pi is equal to CRT, molarity formula directly दबाओ आपको मिलेगा. And which of the following conclusion could be derived from Rutherford alpha scattering particle? So not, see, this is a trap. NEET examination, physical chemistry, Ria, only theory, maximum theory will come. In the overall paper for chemistry, I can expect 5 numericals maximum. 5 numericals maximum. Happy, Ria? That is my perception. Maybe 1 or 2 like this. So physical chemistry means don't say it is completely numerical. So these are the paper. Yes, okay. And it is not. Yes, no. So negative sense the question. Electron move in a circular path, fixed energy called subshells. What is the word called subshell is not related to Rutherford. That is the answer. Okay. And also helium atom and hydrogen atom molecule are moving with the same velocity. Lambda is mv. So helium and hydrogen moving with the same velocity. So velocity of hydrogen, okay, and mass of hydrogen and velocity of helium and mass of helium. Now you take these two can cancel. So this is going to be 1 by 4, 2 by 4. Molecular mass of hydrogen is 2 and helium molecular atomic mass is going to be 4. So the answer is going to be 1 is to 2. 2 by 4 is equal to 1 by 2. Understand? Simple. Do you feel is a numerical? Riha? Not at all. Okay. Now, set of molecules violates the octet. Third period elements violate octet. More than that. So, violate octet, violate octet, violate octet. It is not violating octet. So, the answer is going to be 1, 2, 3. 1, 2, 3. Next, match the example. KCl, you have pure ionic bond, and H2, you have pure covalent bond. K2SO4, you have both ionic bond and covalent bond. Copper metal has metallic bond. Simple application of your metallic and ionic and covalent bond. Okay. And talking about the so in the following reaction, which one gets reduced from reactants? So it is going to be +6 oxidation state. It is going to be +3 oxidation state. Decrease in the oxidation state is called reduction. Chromium is getting reduced. Fantastic. Now, so you see how I am solving the paper. It is simply swift and speed. So for NEET examination, the game is speed. The game is speed and accuracy plays a crucial role. And sulfur, oxygen gives us sulfur dioxide. Sulfur dioxide, oxygen gives us sulfur trioxide. Find the heat of formation of sulfur dioxide. To form this heat of formation of sulfur dioxide, I want to make this in the reverse so that sulfur is going to be striken off. So here sulfur trioxide should be striken off, right? So I just want to say SO3 gives rise S plus 3 by 2 O2. Okay, heat of formation, right? And it is reversed and it is going to be SO2, no? S plus 3 by 2 O2 gives rise to SO3 and 2x. Carefully see. Delta X, delta H is equal to minus 2x. Mind it, it is given like this. It is minus 2x. Product is forming the energy. So reverse this. Second one, SO3 gives rise SO2 plus half O2. Okay, delta H is equal to plus Y. On solving, so you're going to see SO3 is striking off. Okay, and on doing S plus O2 gives rise sulfur dioxide. Delta H is equal to minus 2x plus Y. So answer is going to be this one. Okay, and it is going to be one. Next one, which of the following has delta S greater? Delta S, once this one, A plus B gives rise to C plus 20 kilojoules, 2 kilojoules. Thermochemically, it is written as negative exothermic. Na? It is written outside the equation. It is going to be 2 kilojoules. You understand? Clear? Yes, exactly. Now, which of the following? Delta S is greater. Entropy is increased, Ria. When entropy is increased, na, solid, gas, solid entropy is decreased. Okay, aqueous entropy is decreased. So, it is dissolution entropy is always positive. Mansa, entropy is always positive. Paper will come like this only, na? Yes, high beta, randomness increased. Okay, paper standard and twist will be there. You just understand the twist, don't go exaggerated way. Okay, next, for the following some statements about this Kp and Kc, you have to identify the correct. Kp has always units, of course it has units. Kc has no units in all times, yes, of course. And if delta n is equal to 0, Kp and Kc have no units, no, some units will be there, either atm or mole liter inverse. Okay, and no. Okay, here it is going to be Kp has always units, it cannot be, and correct one is this one. This is wrong and this is wrong. Okay, triple four, wonderful, clear. Right. Next question, the volume of 0.1 molar sulfuric acid required to neutralize KOH react with H2SO4 gives rise K2SO4 plus H2O. On balancing it is coming to, so M1V1 by N1 is equal to M2V2 by N2. Okay, so let us say this is N1 and this is N2. Okay, so the volume 0.1 molar and it is going to be 250 ml of 250 into 0.1 by 2. So on solving, volume is going to be answer 125 milliliters. Right? Easy peasy. Yes, of course, Manasa. And number of H plus ion present in 10 ml of solution of pH is equal to 3. pH is equal to 3. Mean to say pH is equal to negative log to the base 10 of H plus ion concentration. H plus ion concentration is equal to 10 to the power of minus pH is equal to 10 power minus 3 mole per liter. Mole per liter. So 110 ml में कितना होगा? Okay. So that is going to be 10 power minus 3 into 10 milliliter into Avogadro number. So that many are there. So when you solve it, you're going to get this. Understand? Clear? Easy peasy, no? Understand? Next, KSP CR OH thrice gives rise Cr3 plus 3 OH minus, and KSP value is equal to Cr3 plus OH minus cube. Okay, and this is going to be S3S, so you can say S3S cube. Okay, and 27 KSP S to the power 4. So you can calculate S is equal to KSP by 27 to the power 1 by 4. KSP is given 1.6 into 10 power minus 30 by 27 to the power 1 by 4. Understand? So the answer 1. Fourth root is given now. Right? Perfect. And the correct order of acidic strength. Conceptual. HF, hydrohalic acid, water neutral, ammonia basic, and it is nothing. Right? And given below two statements, A, D, E, K are fat soluble vitamins, therefore it is stored in the liver. So that's good. Both are correct statement and it is a correct explanation. So fat soluble vitamins are stored in the in the liver. You know the biology students well. So which of the following postulates of Werner theory? You have primary valency and secondary valency. It is ionic and it is coordinate covalent bond. Okay, so it is primary valence is ionic, primary valence is equal to coordination number. It is wrong. Understand, Manasa? Right. And which of the following coordination compound is the bridged carbonyls? Carbon monoxide, cobalt, it is linked with the carbon monoxide. It is having bridged carbonyl system and it is going to be CO and CO and it is going to be CO octacarbonyl cobalt. It is going to be the first option is the correct answer. That's good, wonderful. Ashish, hi Ashish, right? It was a bio question, Riya. Ashish, Manasa Chaudhary, come on, next. Which of the following can differentiate benzaldehyde? This is your benzaldehyde, okay, and ethanol, CH3CHO. Okay, let us see here. Tollens, Fehlings, and haloform. When you see this, tolerance is given by this as well as this. Fehlings is not given by this. Haloform is not given by this. So check this answer here. One is given, another one is not given. Then only you can differentiate it. Understand? Clear? So that analysis you should have option 4, 2 and 3. And incorrect statement. Which of the following is incorrect statement? PbI4 is highly stable. How? So due to inert pair effect, Pb plus 4 is not stable and I minus is a strong reducing agent, so that this is going to kill. So these two cannot stay together, so that it is not stable. Answer 4 is the correct answer, right? And here you can see ammonia carboxylic group, you are going to get C double bond O NH2 and C double bond O NH2. So Br2NaOH, Hoffman bromamide reaction, so that this is eliminated, this is eliminated. So I am going to get amino group on the either side. Hoffman bromamide reaction. So my answer for this question is going to be 3 is the correct answer. Getting or not? That's good. Next, which of the following is not a correct statement? In this, yes, XCF6 has distorted tetrahedral. How? Not correct. Not correct. Ashish, simply you can find it, right? Rest of the things are given in the NCERT and these are the NCERT points. As it is given, and of the following show minus I effect, except all functional groups show minus I effect. There is no exception. So this O minus is positive inductive effect. Okay, this is positive inductive effect. Next, the hybridization of central atom MnCl6 4 minus. So it has Mn plus 2. Cl is a weak field ligand. So Mn4 2 plus is going to be D5 configuration 4s0. So D5 configuration 1, 2, 3, 4, 5. So it is going to be 5 and outer orbital weak field ligand na outer orbital complex sp3d2 and 5 unpaired electron. Are you getting or not? Perfect. And which of the following liberate CO2? Alcohols do not liberate, phenol do not liberate, carboxylic acid liberate. So answer is going to be 3. Carboxylic acid liberate. Phenol do not liberate carbon dioxide from sodium bicarbonate because the carbonic acid is weak acid as compared to the phenol, right? And which of the following give the positive test for 2,4-DNP? 2,4-DNP positive and negative test with tolerance. Negative test with tolerance and can react with iodoform. Right? Can react with an iodoform, right? That's good, perfect. And one more, which of the following order is not correct? This is important. Please identify yes, no in this, yes, no in this criteria. Okay. Now in this case, bond dissociation energy galat diya. Okay, Cl2 greater than Br2 greater than F2 greater than I2. Okay, that's good. Hi. And 82 grams of sodium ethionate, CH3COONA when it is electrolyzed. Okay, so it is going to give you ethane C2H6 and you are going to get sodium hydroxide plus hydrogen plus carbon dioxide. So the liberated Y and the gas at cathode. So it is going to give you 1:1 ratio. Okay, that's good. 1 mole is formed, right? And it is going to give 1 mole of this. Understand? 1:1. 2,4-DNP test RIA, 2,4-DNP test can be given by any aldehyde or ketone, any carbonyl compound. RIA, fine? Okay, next RONA. Okay, it is going to participate yes N2 reaction to gives rise ether. This is what is called as a Williamson ether synthesis. So in this Williamson ether synthesis, it's a nucleophilic substitution reaction, correct? The alkoxide of sodium may be primary, secondary, or tertiary. It is correct. The better results to obtain the primary alkyl halide because it is participating in SN2. It is used to prepare only symmetrical ethers. Unsymmetrical ethers can also be prepared, but you cannot recall it. Okay, that could be better, right? Yes, this. And which of the following compound does not have sp2 hybridized? Simple acetonitrile, CH3, C triple bond. This is called acetonitrile or ethane nitrile. Okay, so this carbon atom is sp3 hybridized, this carbon atom is sp hybridized. Understand? Okay, next, which of the following is not? Again, entire question paper is like this. Okay, Ashish, बोलना है क्या? Not the property of interstitial compound. Interstitial compounds are highly inert, chemically inert. Chemically reactivity is very high. It is not correct. They are inert. Okay. That's good. Which of the following cannot be detected by isocyanate test? RNH2 primary amine react with chloroform and potassium hydroxide, alkyl isocyanide is formed. This is called isocyanide. Isocyanide. Okay, this is called carbylamine. This is given by only primary amine. So primary amine it can give, secondary amine will not give. It is a primary amine can give, primary amine can give. Don't look at the degree of carbon, look at the degree of nitrogen. Exactly, only primary amines it give, right? So I want to say BB lock, Ashish. Next, arrange cerium, lanthanum, prometium, yttrium among lanthanum 3 plus is bigger than, cerium 3 plus is bigger than, prometium 3 plus is bigger than, yttrium 3 plus. So you have to follow lanthanide contraction. If you in detail, I can explain in detail. Lanthanide contraction you can use, Ashish. 171, no? Okay, sure, definitely. This is called primary amine. This is ammonia. This is ammonia, right? If ammonia, one hydrogen is replaced by an alkyl group, that is called primary amine. In ammonia, okay, two are replaced, that is called secondary amine. In ammonia, 3 are replaced, that is called tertiary amine. Understand? Now look at this primary amine, this should be present. This is present, primary amine. This is present, primary amine. This is present, primary amine. NH is secondary amine. Okay, NH is secondary amine. Understand? Ashish, fine, happy? This is 171 only. Ashish, say happy. Yeah. Now let's move. And C6H5CH2Br, C6H5CH2Br, when it is treated with magnesium in dry ether, you are going to see CH2MgBr, and further it is hydrolyzed, so that H2O H plus. So Grignard reagent is going to be hydrolyzed. Okay, so what is the nature of Grignard reagent? This is the nature of Grignard reagent. Grignard reagents are strong base, so it can react with H plus and OH minus like this. So we're going to see toluene is the exam, the answer. So it is going to be Mg, Br and OH. So answer is going to be toluene is the answer. Correct? Perfect. Lovely. So next question. The correct order of decreasing acidic strength. Trichloroacetic acid, CCl3COOH is greater. Then trifluoroacetic acid, CF3COOH is greater, and acetic acid and formic acid, HCOOH is greater than CH3COOH. So that is the order, and I want to give you a key point here. Acidic strength is directly proportional to negative inductive effect and positive inductive effect. That is the key point. Acidic strength increases, Ka value increases, pKa value decreases. Mind it, Manasa and Ashish. Now let us see the number of structural isomeric amines that are possible for C3H9N. So that CH3, CH2 and CH2NH2, CH3, CH and CH3NH2, CH3, CH2NH and CH3, and N, that is going to be CH3 and CH3 and CH3. Check the molecular formula. I got 4. Wonderful. One more question. SF6 is a gas, SCF4 is a liquid, and TF4 is a solid. Sulfur halides of group 16. Yes, it is true. Among hexafluorides of sulfur, SF is the most stable because of its symmetrical octahedral. So both statements are correct. Highly stable. It is highly stable. Understand? And dipole moment is 0 for this as well. So answer 1 is the correct answer. Next, it is going to be transition elements. A graph is given NCRT. Vanadium is the element with highest enthalpy of atomization. Graph is also given. Mind it, you just see. Okay. And NCRT. Stick to the NCRT, just go through the graphs once. Among the following example, electrophilic substitution. Benzene participates in electrophilic substitution reaction. Nitration is an electrophilic substitution. Okay, coupling reaction is also electrophilic substitution. Friedel-Crafts reaction is also electrophilic substitution. Sandmeyer reaction. What is Sandmeyer reaction? This is your Sandmeyer reaction. N2Cl. When it is treated with Cu2, Cl2 and HCl, you are going to get a chlorobenzene, and here this Cl minus is going to attack here. So it is nucleophilic substitution reaction, not electrophilic substitution reaction. That's very important. Okay. So the answer for this question is going to be 2. And the element with atomic number 111 element is Roentgenium, Rg. So it is there. Named elements, scientists named element: Mendelevium, borium, rutherfordium, curium, Roentgenium. These are the elements which is very, very important. You just memorize from the periodic table in the NCRT textbook. My dear children, is very important. And which are the most react towards SN1. I want to give you a trick before the examination. So running with the questions is different. Ashish, come on. Reactivity of SN1 is directly proportional to stability of carbocation. And this is one trick, 1 + 1 offer. And I want to give you rate of SN2 reaction is inversely proportional to steric factor. So this is the number 2. Understand, clear? Now when you see this forms a carbocation, C6H5 is going to be C6H5, and it is going to be CH3 plus. And answer 3 forms the stablest carbocation is very, very, very important. So we are done for the day in a rapid fire round of the chemistry, 45 questions. Okay, well, you all did, and nice interaction in the series of grand test paper discussion with you guys. And we are done from our side. And last minute, just go through your examination, just a smart revision if possible, and take care of your health. Stay hydrated, eat nutritious food, light food, and have a healthy sleep. Don't think anything. In overthinking also creates a depression. No, just chill and talk to the family members. And you are about to celebrate your preparation harvesting day, that is your examination. So chill in the examination. All the best. God bless you all. Signing off, Suresh, Faculty of Chemistry from Infinity Lane. For esterification, we check the carbocation. For esterification, you check the steric factor. Okay, more is the steric factor for the carboxylic acid, less is the esterification, ease of esterification. Okay, ease of decarboxylation, ease of dehydration. You have to see the order. Okay, take care. Arpita Gupta. Okay, see you guys. Signing off. Suresh, Faculty of Chemistry from Infinity Land. Thank you.